Thank you very much, Joel. I was going to say thank you for the poetic introduction, but then we got down to business. So basically, I'm to give a sort of a kickoff on the background and contextual information. And I think that we are in a very interesting time frame. I don't think this is working. Oh, okay, good. We are in an interesting time frame with biosimilars, and I think we are a little bit behind, and it would be nice to be sure. So am I supposed to aim this somewhere else? Because it's really not working. Okay, well. Essentially, we have the BPCIA, or the part of the Obamacare or the Affordable Care Act, which was really the first way we could define biosimilars in the United States. And it's a biosimilar which is highly similar to the reference product, should not have any clinically meaningful differences in terms of safety, purity, and potency, which is how the FDA regulates products. It must use the same mechanism of action for the conditions of use prescribed, and the same route of administration, dosage form, et cetera, expected to produce the same clinical result in any given patient. And I think that if you really think about how a biosimilar will be regulated, if it is a regulated biosimilar, there really should be confidence that it is going to behave very much, if not exactly like the reference product. Now, there are a lot of approved biosimilars. We know that EMA has been having this ability to approve a product as a biosimilar for a lot longer than the United States. And you can see that this has been followed by many other countries, including Canada, which actually approved a biosimilar to Remicade, infliximab, even before we did. And it's been out in use in Canada for the past year. And that's of some interest, since Health Canada has often been the follower of the FDA, and they found themselves to actually be to precede the FDA on that decision. So I really can't use this. I have to hit it 3 or 4 times before it's working. I hope we have something else to use. So I just wanted to remind you that biosimilars are very different from what we would call intended copies or biomimics. And we have quite a few of those available around the world. We in California concern ourselves about this a little bit because, in fact, our patients might go down to Mexico and could find a biomimic. And in fact, cacuzumab, which was a intended copy of rituximab, was actually taken off the market because of repeated anaphylactic reactions. So these are products that may have started out to be very similar to the reference product, but they've not been through the regulatory pathway. They have not been stringently reviewed and essentially do not, in fact, meet the criteria to be a biosimilar and should be of concern. And of course, they have also been very important because many of these are made in places like Shanghai, India, and Mexico. So biosimilars must be systemically engineered to match the reference product. We can see that at the bottom of the pyramid is what's the most important part about characterizing biosimilars. If they are structurally, physicochemically, biologically, and functionally highly similar, then there will be relatively limited toxicology because there was relatively limited toxicology of the reference products. Clinical pharmacology will be important because of PK and immunogenicity. And finally, there's usually only 1 or 2 clinical trials, and thus the efficiency of a biosimilar program, so that in fact it is developed and approved relatively less expensively than the reference product. So the challenge with biosimilars is knowing which differences do matter clinically. We can start off with something like aspirin, which is very simple. We can go to insulin, which is still considered to be a relevant biosimilar in Europe, but not in the United States, where the definition is more than 100 amino acids. We have somatropin, which was actually approved in the US, but not through a biosimilar pathway, although it will revert back to that. And then we have the monoclonal antibodies and soluble receptors that we rheumatologists are most familiar with. And as you can see, they are far more complicated. And we have to understand their protein structure. So the primary structure has to be identical to the reference product, and that's actually known and patented and in the public domain. But the secondary, tertiary, and quaternary structure is where you can introduce some variability through the manufacturing process, although you want to make sure that whatever variability is there is not going to have clinical relevance. And that's a very important point. And part of that is also because what happens in manufacturing is post-translational modifications. And such things as glycosylation, lipid attachment, phosphorylation, these kinds of things can change through the cell expression system and the manufacturing process. Now, small changes we would consider to be drift. Large changes we would consider to be manufacturing changes, and those are obviously intended. But the fact of the matter is that these small post-translational modifications can change things, such as membrane localization or activation, or even the half-life of the product. So if we look at the history of the commercial batches of etanercept as an example, this was published by work from Sandoz, that you can see that the analytical methods that are looked at to look at a series of lots of Enbrel over time showed, in fact, that there was a manufacturing change. We don't have another one of these? Thank you. So that essentially, right after about 2010, 2011, you can see that a major change occurred. And in fact, that was associated with the building of the Grange plant outside of Dublin for ex-US manufactured etanercept. And that, when you build a brand new plant, you actually are going to have major changes in manufacturing. Now, we would say then that because of that kind of change, we would have data from clinical trials to show that the product from the new production was not different from the old production. Well, in fact, there probably was a clinical trial, or shall we say it was embedded in an ongoing clinical trial. But all of this data is confidential. It's confidential between FDA and the sponsor and remains that way. In Europe, the EMA will make a public announcement, but the details and all of that still remains confidential. A similar thing happened with commercial batches of rituximab, and I know you can't see this very well, I think because of the window. But again, there were major manufacturing changes, and you can see that actually these changes resulted in rituximab being more effective in mediating ADCC and CDC, which is the underlying mechanism of rituximab. Now, this was associated with Genentech building 2 plants outside of Vacaville, California, to produce rituximab. And in fact, the new manufacturing plants were so efficient that they actually closed down one of them, and they only use one for rituximab, and they use the other for other manufacturing. But again, these are the kinds of things that have happened over time that we know, and we know based on what EMA has announced that there have been multiple changes with many of our products, particularly the ones that have been out for the long time. Some of that is related to having production within the US versus ex-US and building new plants, as I've said to you. A lot of it is about the fact that we've learned how to manufacture more efficiently and better ways. And because of that, these changes have occurred. And you can see that, of course, most of them have been around infliximab and etanercept, but also even Humira. So these are essentially manufacturing changes that are planned upon. And depending on the amount and type of manufacturing change, as you go from a low-risk process to a moderate-risk change to a high-risk change is when you will have to have clinical data to prove. Because no matter how well we can characterize something ex vivo, in vitro, there are times when you would not know that some change could, in fact, alter how a product actually performs clinically. Now, that's very different from what we call drift. And drift is different from an intentional manufacturing change. So what we're looking at here is the upper specification limits and the lower specification limits, and then we're looking at the upper and lower control limits, which are much tighter. And you're seeing here, as an example, that all of these lots fit in with the UCL and the LCL, with the exception of this one lot. Now, that lot would have been rejected. So essentially, it's a very tightly controlled range of attributes that's looked at for every single lot, and they have to fit into those. And thus, the concern about drift is really being controlled for. But in fact, as we've learned to characterize products better, and as we're now making biosimilars to be as highly similar to several, maybe multiple lots of a reference product, in fact, these UCLs and LCLs, these control limits, are actually becoming even tighter with a biosimilar, and they're even more stringent. So essentially, EMA characterizes biologics, and in fact drugs, as quality, safety, and efficacy. The FDA talks about safety, purity, and potency. And then we also have a definition for not just highly similar, but interchangeable, which is actually unique to the US, and I'll come back to that in a minute. But what we realize is that biosimilars are going to have a very detailed chemistry, manufacturing, and control section, because they're actually going to have more analytic data than the reference product, than the innovator. And this is because they've been reverse engineered to match. And on the basis of all of that, and on the basis of what we now know that we can do for characterization, and in fact for reverse engineering, that means that we now have a tighter batch-to-batch variability than we would have had with the reference product. And we actually have more data to characterize the biosimilar. So that's really a very interesting point, because when we think about trying to match a previous product, we're now matching it even more carefully than the product itself to itself. Now, the FDA has issued 9 guidance documents since 2012, when the Affordable Care Act was passed and they could regulate biosimilars. Scientific considerations in 2012, which was actually finalized in 2015. Quality considerations also. Questions and answers. It's a kind of a novel guidance document, questions and answers, and that actually was issued and finalized, and a new additional questions and answers document is now out there. And these are based on interactions with sponsors about biosimilars, updated as appropriate, trying to answer the questions about the process and also the regulatory system. Formal meetings between FDA and biosimilar sponsors. There's a draft. And this is because we have the Biosimilar User Fee Act, which is essentially funding this work in the context of the regulatory process. And then clinical pharmacology data. And this was a very unique guidance document, because it was the first one that said, no, you don't have to just compare this to a US manufactured reference product. We want you to compare it to an ex-US manufactured product as well as a US manufactured product. And some have said that an ex-US and a US manufactured reference product may actually be almost like biosimilars of each other. And thus now you have a 3-way comparison that is actually even more, shall we say, stringent, but also useful clinically. And then we have reference product exclusivity, which meant that you could not apply for a biosimilar application, which is a 351K, as opposed to a new BLA, which is a 351A. Can't actually apply for it until at least 4 years after the reference product was licensed, but it can't be made effective until at least 12 years. Of course, that doesn't have a lot to do with the patent issues that come to play, but also if you do pediatric studies, you may get 6 months of exclusivity. Now we have the non-proprietary naming draft that came out in August of last year. Essentially, the proper name is used with a designated 4-letter suffix. Now, the proper name isn't always the generic name, but it is the name that we usually use for the biologic product. The 4-letter suffix is essentially based on also what WHO has recommended, but these are supposed to be actually done on a basis of no resemblance to any name that we know of. And thus we have some very interesting 4-letter suffixes that may be relatively difficult to remember and may be hard in the context of pharmacovigilance. So it should be interesting to see what happens. These are all generated randomly. And so we have, as of recently, we have DYYB, and we have SZZS, and we have ATTO. And I'm not sure any of us are going to really be able to understand what those particular suffixes mean. And then labeling, which very similarly to EMA regulations, means that the label is going to talk about the reference product. It is not going to talk about the biosimilar. And if you want data about the biosimilar, you're really needing to read what has been published about the trials with the product. And it hasn't been a lot published about the preclinical characterization, et cetera, but I think we'll start to see a little bit more about that. But we certainly have seen, at least in rheumatology, publication of the papers in rheumatoid arthritis and PK and immunogenicity and so on. And that is really where you will find it. We're also told that you can look in the Purple Book online, which the FDA makes as a list of all the products they've ever approved, but it doesn't actually tell you anything about the product. So the demonstration of biosimilarity is actually similar to, but different from changing a manufacturing process. But we're using the same technology and techniques to do that. So comparability exercise for manufacturing process change is above, and then the demonstration of biosimilarity is below. So the difference, of course, is that we don't know the innovator's process and manufacturing history. We know the primary structure, we actually know the type of cell line that it's made in, but we don't have any other knowledge of it beyond that. And then the analytical studies and non-clinical studies, and finally the comparative clinical PK/PD studies and immunogenicity are what go into defining the biosimilar. But we're using a lot of the techniques that we developed when manufacturing process changes were done and had to be characterized carefully. So we're essentially starting with the known DNA sequence and coming out to match the known clinical data with all the other things in the box as essentially the unknowns to reverse engineer a biosimilar. So it's really a very interesting technique and technology. And as I said, it's based on the 20+ years we've had of manufacturing biologics, and which have taught us a great deal. So essentially, if you look at what's required for an originator biologic, we start off with a characterization and spend a lot of time on phase 3 clinical trials, often with 2 studies in each indication as replicate studies. When we turn this around for a biosimilar, then we see that we spend very little on the clinical studies, 1, maybe 2, and the majority of it is the physicochemical and functional biologic characterizations. So that's the difference. But we have to do very, very careful analyses, and so they've been described as fingerprint analyses. Essentially, for a monoclonal antibody or a fusion product, they look a bit like this cartoon. We start off with a primary structure and the attributes that are related to the amino acid sequence, the post-translational modifications that I talked about. We look at the secondary, tertiary, and quaternary structure, which can affect function majorly. The receptor binding and immunochemical properties, in other words, the biologic function. The quantitative levels of product variants and identities, which can be introduced during the process or subsequently. So they can be degradation profiles as well. So product-related substances and impurities and stability, the process-related impurities, and even the impurities that can arise from the host cells. So particles and aggregates, and also this is all washed out over here, but strength and formulation. So essentially, we have tests that confirm the high degree of similarity in the process and also in the behavior of the biosimilar. And we call them CQAs, or those that are considered critical quality attributes. So for instance, for a TNF inhibitor, they're related to how a TNF inhibitor would actually function. So the amino acid identity, both binding to soluble and transmembrane TNF, and in vitro neutralization of TNF are considered to be some of the CQAs that we would look at for these type of products. So essentially, if you look at a monoclonal antibody, you could talk about what the CQAs would be generically in terms of how the product's put together. So we actually know that the FC-mediated functions are not important for the biologic function of the product, but they are certainly important for the half-life and the clearance mechanisms. So I can't really actually show you all this because this window is too bright on the slides. I guess you can turn to look in the back. But essentially what we're looking at is functioning of the FAB portion. We're looking at the cell line dependent higher order structure. We're looking at, in fact, TNF neutralization or epitope binding, which is both cell line and process dependent. Cell line dependent for the FcRn binding, which of course is the clearance mechanisms and the half-life of the product. And also the cell line and process dependent contact and product things. So all of these are CQAs that are very important to the product. Others of them are characterization that wouldn't be considered a CQA per se, but would be looked at in a statistical way that was a little bit less stringent. So the clinical trials are supposed to resolve residual uncertainties. And essentially that you see that the product is functionally and characterized as being highly similar, then what we have left as residual uncertainty is a PK, the immunogenicity, and actually how the product behaves clinically in terms of efficacy and safety. So PK is required to be equivalent, and this is equivalency that's actually defined around the world for drugs as well as biologics, as well as biosimilars. So we're looking at essentially an 80 to 125% range for the 90% confidence interval, which is actually more stringent than the 95% confidence interval, so that the maximum serum concentration, the area under the time concentration from first time to last time, and from first time extrapolated to infinity, all have to be within these ratio of 80 to 125%. Then we also need to show equivalence of the efficacy. Now, we can't ask for non-inferiority. It has to be equivalence. So now you have to define an equivalence margin that's based on the reference product. So that's essentially to look at all the clinical trials with that reference product in that specific indication and ascertain what is the effect size, essentially, that will preserve that margin. So we're looking for not really 50%, but much more closely to 75% or so that the lower limit of the 95% confidence interval between the biosimilar product and the reference product do not cross the margin. And essentially, it's also said that if there's meaningful correlation between a PK or a PD marker, that this could be done, and perhaps we wouldn't need to do an efficacy trial. But of course, in rheumatology and most of the autoimmune diseases we've looked at, we don't have a PK or a PD marker that would be able to do that. And so we have to look at efficacy. And of course, we do look at the PD markers as well. Now, the FDA guidance also specifies 2 separate studies of immunogenicity, both pre-market and post-market. And that comes to mind because of the subtle differences that may occur over time. So if we look at the biosimilar clinical trials, we'll see that there's actually some very strong similarities all the way across because there's not a lot of variability that could really be done. We're not thinking about unique trial designs with biosimilars because we essentially want to show that they are equivalent to the reference product. So we can pick 1 or maybe 2 indications. Some manufacturers have picked 2, others have settled with 1, knowing that there will be extrapolation. We can talk about study populations. Is it early disease, advanced disease? Which indication are you going to pick? We can talk about whether it's with monotherapy, where likely you'll see more immunogenicity, or combination with standard of care, which should show less immunogenicity. Also, will there be a single switch, or will there be multiple switches within the study? We'll come back to the primary endpoint, because you want a sensitive endpoint that's going to behave similarly, so that you can count on the fact that your trial will actually replicate the types of trials that have been done with the reference product. And then the duration. We're now doing rheumatoid arthritis trials with a primary endpoint at 12 weeks because we don't want patients to be exposed to placebo longer than that. But these are not placebo-controlled trials. They're active comparator studies. So you can pick longer time points. But it may be appropriate to do switches at earlier time points. So all of these are supposed to be in such a way that the endpoint, the population, the sample size, and even the study duration is going to be adequately sensitive to detect any differences between the reference product and the biosimilar. So essentially what we're looking at is not a superiority study, not where you look for a non-inferiority and then you can look for, in fact, superiority. We're actually looking for equivalence, whereby the biosimilar is similar to the reference product and the treatment differences are squarely between the lower and upper margin of the definition within a 95% confidence interval. So examples of biosimilar clinical development programs, well, healthy subjects have now been used typically for the 3-way PK study, which is of interest because some people have worried about the ethics of actually exposing healthy subjects to a monoclonal antibody or a soluble receptor with the idea that perhaps someday they will need a biologic for their treatment of their subsequent autoimmune disease or their cancer or whatever, and would a single administration of a non-relevant monoclonal antibody have an effect on subsequent immunogenicity? Well, probably not in terms of epitope binding, but we do know that some products do elicit antibodies to the Fc portion, and that might become very relevant. Rheumatoid arthritis has been one of the very common indications to look at, but so has plaque psoriasis. And both of these have been the ones that have been picked most frequently by our biosimilar manufacturers to date. And part of that has to do with what the outcome measurements are and how well they perform. So the most sensitive models have been basically based on ACR 20, 50, and 70 and PASI responses because, in fact, they have behaved most similarly on a case-by-case basis across multiple trials and multiple products. And the other point is that we also know that there's differences in immunogenicity. So we know that rheumatoid arthritis and psoriasis are characterized by fairly high levels of immunogenicity, whereas we know now that spondyloarthropathy actually tends to have a much lower set of immunogenicity. I'm not sure we know why, but this has been clearly shown with the infliximab biosimilar study in SPA versus rheumatoid arthritis. There's also the issue about safety and extrapolation. Extrapolation is a major issue in terms of taking data from RA or psoriasis and extrapolating it to IBD. But unfortunately, our IBD outcomes do not behave as well as our rheumatoid arthritis and psoriasis outcomes do. There's been several trials that have failed on the basis of looking at a CDAI, and in fact, the Mayo Clinic response also doesn't behave well in ulcerative colitis. And then do you want to stratify according to baseline characteristics? Well, we didn't do that in the early days of the reference product trials, but there's certainly a reason to consider it now. We know the effects of BMI, for instance, and we know that we have different doses recommended according to BMI. We've often looked at CRP, and we definitely know that there's differences in responses based on how many DMARDs or biologic DMARDs a rheumatoid arthritis patient has failed. And then there's a lot of requirements for many of these studies to have patients be biologic naive and specifically TNF naive. But that means you're going around the world to find all these patients because it's getting increasingly harder to find bio-naive rheumatoid arthritis patients. So, for instance, here's some global efficacy trials for the adalimumab biosimilars. You can already see how many adalimumab biosimilar manufacturers there are. And we know, of course, that the Amgen product has now been approved in the US as adalimumab ATTO. But essentially, if you look at those studies, then you also look at, here are the differences between active and placebo response rates. And as you notice, you can see why plaque psoriasis has been actually selected as one of the indications, because that is the largest differentiation between active and placebo response in the adalimumab development program. And also in RA, we see that the ACR 20, 50, 70 nicely differentiate, although you see that there's a better differentiation even with the background methotrexate than with the monotherapy. But essentially, this is a nice illustration of why we've seen RA and psoriasis picked for their clinical trials. Now, the design elements seem to differ depending on who the sponsor is and what the stringency is in the context of whether patients are allowed to have had previous biologic therapy or not, and also how many switches or when the switches occur between, for instance, a reference product and the biosimilar. Different time frames for many of these different trials. But essentially, all of them are likely to be successful. There's probably other reasons to be concerned for maybe differences. Now, with psoriasis, we can also see that we have several different designs for them as well. But in general, most of the sponsors have allowed prior biologic experience in their psoriasis studies. Psoriasis is a good indication because we don't use background therapy, and it has similar immunogenicity to rheumatoid arthritis, so it might be the example of the worst case immunogenicity with a product as opposed to RA with background therapy. But again, the transition designs can occur at different time points. Typically, though, again, 12 or 16 weeks is when the primary studies were differentiated, and that's when you can do the switch in these studies. So now we have the example of the first biosimilar that was actually approved in the United States, but also in Europe in terms of a monoclonal antibody, and that was the infliximab biosimilar. It goes by lots of different names. For because it essentially has multiple different sponsors selling the product, although all of it is made by Celltrion in South Korea. And Celltrion is also known as a good contract manufacturing organization for multiple reference products. So Inflectra essentially is the name that it will go by in the United States, although it's infliximab DYYB. So essentially, it was approved in April after a February 9th Arthritis Advisory Committee meeting that had GI members, et cetera. And it was an interesting vote, 21-3 to approve it. So this is a PK profile following a single dose IV of this product versus both ex-US and US manufactured infliximab. And you can see that essentially these are very highly similar to each other. This is after steady state. There's been an argument that you should look at it while you're reaching steady state, but in fact, if you want to look at comparability, steady state is the appropriate time to look. So I think you can't see this, it's washed out, but this was the Planet AS or Planeta study in ankylosing spondylitis, which was used at first as a PK, but also a secondary efficacy study. Subsequently, the Planet RA or Planet RA study, which was looked at for essentially the ultimate clinical trial in rheumatoid arthritis with background therapy with this product. And the immunogenicity was interesting because essentially it showed that there was very similar immunogenicity, anti-drug antibodies, in incidence between both the biosimilar, which was known as CTP13, and infliximab. And this was despite background methotrexate. And this is actually similar to what was published in the EPAR in Europe for infliximab of about 40%. Now the assays are much more specific and much more sensitive now, but essentially they're showing the same type of immunogenicity and should show the same type between the biosimilar and the reference product. And that means also that you should be seeing similar hypersensitivity and infusion reactions. Now we talk about highly similar efficacy. About the most we can do for safety is to say that it's comparable, because these studies are really not large enough to show something that would be considered to be similar, so comparable probably works better. But the other part of it is that immunogenicity should really be fairly similar, although we've learned from these biosimilars that there are some differences in immunogenicity from what we knew before about the reference products. Now this is just to show the same biosimilar, now known as Remsima or Inflectra, and to show that the immunodominant epitopes between the biosimilar and the reference product are essentially the same ones. First, from an in vitro study in IBD patients in Israel, taking patients that had already been exposed to infliximab and did have antibodies, as well as those who had been exposed but did not have anti-drug antibodies. And essentially, they show that these epitopes were the same. They did not cross-react with any other monoclonal, such as adalimumab. And in fact, this has also been shown now, publication in Europe, where patients were actually assayed and essentially showed that for all intensive purposes, you can use the same assay for the reference product or for the biosimilar, and you should be able to. You don't really need to have two different assays. Now here's the etanercept biosimilar, which is known as Brenzys in Canada, Benepali in Europe, and is still under review here in the US. But this was a double-blind, multi-center trial, methotrexate incomplete responders. And what was interesting is that they showed less immunogenicity. So the Samsung Bioepis biosimilar had immunogenicity of about 1% versus about 13% with etanercept using a very sensitive assay. And this sparked about 8 different letters to the editor in Annals of Rheumatic Disease about why did this happen, were there differences in formulation, et cetera. Well, in fact, there were not differences in formulation. That is not what happened here. What happened here is that etanercept is a very interesting product, and it essentially has differences in the way that the portion, particularly of the Fc receptor, but some of the, or Fc portion, but some of the other part of the molecule where they can actually mislink or miscrosslink when they're sitting in vitro and sitting ex vivo. But when actually put it back into man or into serum, it all resorts in the same similar way. And so presumably some of this is part of the reason for the variability in the immunogenicity assay, but it actually is not clinically relevant. And what we see here is that these differences were relatively small. Now, we have also the ABP 501 adalimumab biosimilar, which is now approved in the US. And what it's shown us is the immunogenicity of itself as well as the reference product. So that in RA with background methotrexate, the immunogenicity is about 38%, and neutralizing antibodies occur in about 10% of patients. When we now look in psoriasis without background therapy, we now see that in fact the immunogenicity is more like about 50% or maybe even 60%. But again, the neutralizing antibodies is somewhere about 9 and 13%. And so essentially what we've learned is with these more sensitive assays, we see more immunogenicity. Is it clinically relevant? Does it matter on a patient-to-patient basis? And I would argue probably not. We know when patients are losing response. But we also know that patients who are anti-drug antibody positive may still have very good efficacy. And we really don't know whether the anti-drug antibodies, even though they may be affecting the clearance of the monoclonal or soluble receptor, they may actually be helping to take the monoclonal or soluble receptor to a site of inflammation in the context of lymphatic recirculation, et cetera. And so although these things matter on a mean basis in terms of a group mean, on an individual patient basis, it may not be relevant. And thankfully, at least in rheumatology, we can monitor well, so we know when patients might be losing efficacy. This, of course, is an issue when we talk about IBD, where it's a little less easy to monitor for loss of response, and thus our colleagues are doing much more in the way of assays for anti-drug antibodies and even serum levels. So there are a lot of biosimilars in development. Some have now been approved in terms of there's at least 3 different infliximab ones. We've got quite a few rituximab ones in the pipeline. We have at least 2 etanercept ones that are approved, and lots of adalimumab ones in phase 3, as I showed you. So essentially, the FDA approved the adalimumab biosimilar and the etanercept biosimilar with extrapolation to all the other indications based on 2 meetings, one in July with the adalimumab and the other the next day with the Sando biosimilar to etanercept. And both of those Arthritis Advisory Committee members, both of those meetings voted unanimously for approval for both of the products, and they were approved as etanercept SZZS and adalimumab ATTO about 1 and 2 months later. So the process is moving quickly now in the US, and we have every reason to believe we will see a lot of biosimilars. The question is still going to be, we have biosimilar A and biosimilar B, and we'll likely be anticipating switches between A and B. Now, they've both been actually developed to be highly similar to the reference product, but are they going to be different from each other? Well, the answer is they're going to be different from each other, but in very, very subtle ways. It's probably very likely that we don't have to be worried about it. But there's still the theoretical concern about switching between multiple biosimilars and back and forth, and I think that that remains a question that we will have to see. But I wanted to stop with the NOR-SWITCH study, which just was announced, the results about 10 days ago. And you may remember that this was funded by the Norwegian government based on the fact that Celltrion deeply discounted their biosimilar in Norway. It was a non-inferiority study where they enrolled approximately 500 patients who've been receiving infliximab for RA, spondyloarthropathy, psoriatic arthritis, ulcerative colitis, Crohn's disease, or plaque psoriasis. And essentially, they were enrolled, they'd been on stable treatment for at least 6 months, and they were randomized blindly to either continue the infliximab or to switch to the biosimilar. And the primary endpoint was disease worsening. So this was designed to answer the question that we've really wondered about. And then once they completed 12 months, they were asked to enroll in an open-label 26-week further study where everybody was switched then to the biosimilar to see whether there was additional data on switching. So essentially, here are the patients. For the final analysis, it was essentially 481 patients. You can see that they were mostly female, or mostly male, actually. The disease duration tended to be long duration, as we know from patients who actually get eligibility for biologics. Many of them had been on infliximab for 6 to 7 years, which is very interesting. And about half of them were on immunosuppressive medication, as you might expect. Overall, 30% had Crohn's disease, 20% had ulcerative colitis, about 20% had a spondyloarthropathy, about 16% had rheumatoid arthritis, 6 to 7% had psoriatic arthritis, and about 7.5% had psoriasis. So here is actually the results. Disease worsening was defined according to the disease. Now remember, this study was definitely not powered to look at individual diseases. And so there are variability, but overall, in the entire group, what we see is that the non-inferiority margin was 15%. And in the 394 patients who could be analyzed, the data for treatment failure fell squarely within that margin between minus 12.7 and 3.9%. Now, if you scan down here, you'll see that there are some differences here that look to be maybe some disease-specific differences. But overall, you see that none of these confidence intervals would suggest statistical difference. So essentially, the biosimilar infliximab is now approved in multiple countries, and it was the first to be approved in the US as a biosimilar. We know that the trials have shown that it and other biosimilars have been considered highly comparable to the reference product. They've been broadly extrapolated to all indications, and there's been more than a year's experience in Canada with the infliximab biosimilar, and the provinces who've decided both policy and reimbursement may well serve as a model for our managed care organizations. For instance, in British Columbia, there's about a 50% discount. In Quebec, essentially, the two products are identical in price. So it's very different to see what has happened here. We now have adalimumab and etanercept approved, but we don't know when we will see those, as they are still being held up in litigation, as the infliximab was. We wonder about interchangeability. We've been told that there won't be an FDA guidance on interchangeability for at least another year or so. Interchangeability would mean that someone could get the biosimilar without knowledge of the healthcare provider. Now, already many state legislatures have passed laws to say that that won't happen. And because there really isn't interchangeability anywhere else in the world, I'm not sure that this would ever happen in such a way that we wouldn't know about it. But it has been at least a theoretical concern, as well as an issue about what will be necessary for interchangeability. Because we know when both tocilizumab and abatacept went from IV to sub-Q, they had to study some 3,000 patients with multiple switches and stopping and starting back and forth between IV and sub-Q. Now, if that is what would be required for interchangeability, then I suspect most biosimilar manufacturers would consider that to be not sufficiently efficient and sufficiently expensive to not want to ask for an interchangeability designation. So I'm sure that that's a really interesting discussion that's going on at the FDA right now. We have talked about the naming with the non-proprietary name and the randomly generated suffix. Maybe it will assist pharmacovigilance. Maybe it will make pharmacovigilance a little bit more complicated. We still need continued real-world data on safety and efficacy of switching. We only have the NOR-SWITCH study and some smaller amount of data from the DanBio registry in Denmark. We do have other small series in IBD which have lent confidence to use of the biosimilar. But I think it's really going to be important to get more data, and thus many of us are here on that basis. And finally, I think that one of the really important parts about biosimilars is they have actually increased accessibility. You look at Eastern Europe, and we're seeing patients with rheumatoid arthritis and other diseases who otherwise never would have had access to a biologic. And it's made a huge difference, and it's made a huge difference in terms of accessibility, particularly in countries where the GDP is not as high as we think of here in the US or in Western Europe. And even in the UK, where restrictions to use of products, and particularly biologics, has been very stringent. So it is a new world. I'm not being nearly as poetic as Joel about that, but I think I thank you. Hope this is a good introduction.